the guest speaker, Dr. Manisha Goel, ma'am, Assistant Professor at the Department of Biophysics, University of Delhi South Campus. Ma'am did her graduation in BSc Zoology, Botany and Chemistry from HNB Garhwal University, post-graduation in MSc Biotechnology from University of Roorkee, and PhD from National Institute of Immunology. She later worked as a postdoctoral associate with Dr. R.K.D. Michigan at Bioinformatics Lab, Stovert's Institute for Medical Research, Kansas City, USA. Her lab research expertise includes structural biology, computational biology, and bioinformatics that aim to understand the mechanistics of cellular processes and biological pathways. She was awarded grants under UGC CSIR, DU-DST, ICMR, Rapid Grant for Young Investigator from DBT, and many more. Today, ma'am will be holding discussions with us on CRISPR systems, assigning functional definitions to diversity of Archeal Cas4 proteins for potential applications in genome editing strategies. Before we begin with the session, I would request all the participants to post their queries in the chat box, which will be answered by ma'am at the end of session. Now, without further ado, I would like to invite our guest speaker, Dr. Goel, to grace us with the session. Over to you, ma'am.
Thank you, Kushi. Thank you for the nice introduction, detailed introduction. I'm really sorry about the long topic that I have. The title of the talk became too long, just because I wanted to kind of give you a heads up about what we will be talking about. So basically, as Kushi said, she's already mentioned that my lab works on structural biology and bioinformatics. So by training, I was a crystallographer in my PhD, and then for my postdoc, I moved to bioinformatics. So my lab here, actually, when I joined back in 2010, since then we have been doing a combination of both. And so this is also one of the systems that we are doing the same thing. And so I guess a little bit that CRISPRs are so famous and so well known these days that you probably understand a little bit about CRISPRs, or at least have heard about them. So CRISPRs are basically, but I'll just do a short this one. Let me just start my slides. Let me just share my slides. Sorry, So this is, so it was can you see my slides, Kushi?
Yes, ma'am.
Okay, so basically this CRISPR systems we started working on when I came back from US. At that time, it was just taking off. They had been recognized as innate immunity systems in bacteria and in archaea. So basically, it was realized that CRISPRs were kind of adaptive immune systems in prokaryotes. So basically, a bacterial cell or an archaeal cell that has CRISPRs, that has a CRISPR mechanism machinery, would be, once it is infected by phage, it keeps a part of that phage inserted in its DNA, and then if the same phage and makes an antidote, or kind of say makes a complementary DNA, and then has a whole machinery associated with that. So then the next time when this phage infects the same bacteria, this recognizes it, and it actually goes back and uses this DNA that it has stored in its own genome to go and target that phage, and then cut the phage DNA into pieces, and it is thrown off, and so phage is not able to infect this bacteria. So this is what it does there in CRISPR. So that is why they were known as adaptive immune systems. They were adaptive in the sense that they kind of generate a memory in the cells, and that the reinfection is not easy. So that was, but these were first identified long time back. It took about 20 years for scientists to figure out what it might be doing. So these were repeat sequences. So what happens here is that if you look at the top thing, this is what happens. This is what the arrangement of these CRISPR systems inside the bacterial cells is, that it has a leader sequence, a single leader sequence, but it has repeat sequences. So these are just CG, CG, A, G, A, G, kind of repeat palindromic repeats. Then we have a spacer sequence, and then we have repeat sequences, spacer sequences again and again. So all of these spacer sequences, each of the spacer sequence is what is unique to each phage infection. So in fact, you can actually, if you decode the genome of a bacteria, you can actually figure out how many times it has been, how many different kind of phages it has been infected with, and in what sequence actually. So that was, but it took them a lot of time to figure out what these could be doing, what were these repeat sequences. So basically, repeat sequences are nothing; they're just tags for to be recognized by these genes to realize where to cut them. Now, when we started finding these out, the scientists decoded all the genomes because genome sequencing was available, and it is very easy to sequence the genomes of bacteria because these are smaller genomes. So they figured that there was too much variety in. So this system was not a very homogeneous system. It wasn't like there was only these kind of genes. And then, so there were certain number of genes ahead of this leader sequence, which are collectively called as Cas genes. So sometimes there were 3, sometimes there were 12. And these genes were then realized that they're ahead. So they are all clustered together into a cassette, and then they work together to do everything that a CRISPR function is required to do. Now, so there were scientists who were trying to figure out what is the function of this in the natural systems, but there were other scientists who actually also wanted to figure out how we can use this for our benefit in biotechnology. So then people were able to quickly figure out that if we can use the same system, if we insert this into our systems, whatever cells we want to manipulate or whatever bacterial cells we want to manipulate, we could actually give them a sequence, spacer sequence of our interest, and we could shut down a gene, we could cut a gene, and so therefore we could actually do genome editing. So you could prevent phages being infected from other bacteria. You could actually modify the expression of a gene, and so all of this became very, very interesting for others to. So you could actually change expression. So if there were heterologous systems, you could choose one allele which would be expressed and the other one which would not be expressed, and so that way it became very, very interesting for genome editing. And so that's why even biotechnology companies took over quickly. And so one of the systems, I hope you've heard of Cas9 systems, which are now available in companies. They have ready-made kits for doing Cas9 based genome editings. But what I wanted to do here, so we were one of the labs that we wanted to do the basic biology, understanding what it has evolved, how it has evolved, how does it help the bacteria system, and what is the functions of bacteria, why are they different, why are there different types of Cas genes present here. So basically all the Cas genes that we see here, they were done and they were found to be large group of at least 45 different proteins, but they have similar functions. So sometimes they can all be classified more or less into 7-8 different functions like nucleases, helicases, binding proteins, and some were obviously unknown functions. Now they were kind of discovered as and named as Cas1, Cas2, Cas3, Cas4 in the sequence in which they were discovered, or in the sequence in which they were found ahead of Cas genes, found ahead of later sequences. So basically, then out of a lot of work that went through, it was able to figure out that all the CRISPR systems were not similar. So they were basically divided into type 1, type 3, type 4. Earlier, then it was realized that even within themselves they had differences, and so they were divided into 2 classes called class 1 and class 2. And so class 1 has type 1, 3, and 4, and class 2 has type 2, 5, and 6. And now these two were working slightly differently from each other. So I'll show you what is the specific differences between these two. So basically, this is what it looks like. So CRISPR has 3 steps. So one is adaptation. One is where a virus infects a bacterial cell and an archeal cell, and you keep a part of that DNA in your genome. So that is called as adaptation. Then the expression comes in where the next time it affects, and then you express that part of DNA, and there is the interference where there is a component that takes that part of DNA, uses that DNA to recognize a target gene, in your target gene in your genome, and then tries to shut that down or tries to chew that up. So if you look at it, class 1 and class 2, we have class 1 and class 2. Class 2 is basically having class 2 is basically having long genes. So it is, so if you look at in contrast to class 1 has smaller proteins, so but multitude of proteins, whereas class 2 has one big protein. And so that protein basically does more or less everything. So that is why this system, Cas9 system, became very easy for people to kind of use immediately, because this was single protein that needed to be expressed, and then it could manage everything on its own. So, that is why with this one became a quick favorite with the biotechnology people, with the people who wanted to do genetic mutations. But then the questions about this working still remain. Why do we have other systems? How do they differ from these systems? And there were always some shortcomings with Cas9, saying how do we increase their specificity? How do we change their efficiency? So people kept trying to figure out what do other systems do. So that is where we came in. We were looking at it from the bioinformatics point of view, and we're trying to figure out what do other Cas genes do. So one of the genes that we became interested in over a period of time is called as Cas4. If you see this, Cas4 is actually present in Cas2. Class 2, and then it is also present in one of the types in class 1. So it is here in the adaptation module class Cas4. So it is present there, but it is not always present. So if you look at it in contrast to Cas1 and Cas2, which are always present in this one, always required, essential, Cas4 is present in some systems, some subtypes, but not present in other subtypes. But whenever it is present, it is kind of essential. It gives, it was shown to give specificity. So the question remained as to why do some CRISPRs are able to work without Cas4? What does Cas4 do that it actually is required but is not essential? But it does some other protein take over the function of Cas4 when it is not present? And why is it specific to certain subtypes? So all of these questions remained unanswered. And so this is what we started with initially. So basically, this is what we knew about Cas4. So 1 or 2 Cas4 proteins were crystallized and reported. So basically, this is what we knew before we started our work. So we knew that CRISPR4s were present in CRISPR locus, and they were functioning in the adaptation step. Sometimes in some species, they were reported to be working with Cas1 and Cas2. But Cas4 seemed to be critical for maintaining functional fidelity. But then over a period of time, by the time we were ready to do this, we figured that there was something wrong about Cas4s, in the sense that not all Cas4s that were reported in literature were actually present in the CRISPR cassettes. So if they were not present in CRISPR cassettes, they were actually called as solo. And this is something that was reported by somebody from other groups also. So there were 2 Cas4 structures that were reported, and they were shown to have be nucleases, have 5' 3' exonuclease activity, 3' 5' exonuclease activity. But even among themselves, sorry. So even among themselves, these Cas4s were actually showing differences in their activities, but they had some conserved motifs like 4-cysteines, QXXFI motif, and so we basically assumed that these were the motifs that were helping in function. So as I said, there would be Cas4 genes that were associated with Cas1 and Cas2, and then there were genes that were lying far away from this cassette. So these were basic. Then these people started calling them as solo Cas4s. And then we didn't know, so nobody knew what is the difference between Cas4 of this solo Cas4 or the Cas4 that is found in the cassette. Which of these is working in the CRISPR? Does it have any other functions other than CRISPR function? Is it related at all or not? And among themselves, they have very low sequence identity. So that means they were, so it probably suggests that they were doing something else, or they are doing some housekeeping functions other than CRISPR, if they're lying other than CRISPR cassette. So we actually looked at several Archeal genomes. We started doing bioinformatics, and we looked at about 200, 400 genomes and tried to look at where we would find solo Cas4s. And so we were, the basic question for this project at least was to figure out what role does solo Cas4 have compared to Cas4s that were found within the CRISPR cassettes. Okay, so this was the basic objective of the study. So we wanted to find out how many different types of Cas4 are present, how similar are they to each other, and then we were trying to narrow down on certain Cas4 genes from various Archeal species, do a heterologous expression, purification, and then we would do biophysical and functional characterization to find out what activities do they have, what function they could possibly have inside the cells, and therefore, and then we would want to do crystallization and figure out what kind of, what do they look like, and crystallization was also meant for figuring out their functions. Okay, so that was the objective of the study. I hope that is clear. Am I clear so far to students, everybody else? Anybody has any questions so far? We're good?
Clear, ma'am.
Okay, so let's continue. Fine. So then, so basically, this was the objective. So the objective was just, so objective still remains the same. It is only to figure out, at least for this, so overall objective is to understand how Cas4 contributes to CRISPR function, and if we can actually use Cas4 mediated system as an alternative to Cas9, what advantages would we have if we use Cas4 based systems as opposed to Cas9? Why do cells have different kinds of CRISPR systems? So that is the overall objective, but there is a narrower objective of that in the present project, which is basically to figure out what is the difference between the solo Cas4s and the Cas associated Cas4s. So we basically started with proteomes of Archeal genomes, and so we looked at everything that was available, all the Archeal genomes, about 400 of those, and collected all the sequences, and then did a blast, and then did the classification of those to make a phylogenetic tree to do. So this is basically just telling you what we are going to show the data for ahead. So we already compared it to whatever other people had done, and then we found normal sequences in our data sets, which is expected because the other data sets were a little old. And then so we did a classification of those, and we tried to distribute them into different types. So that is solo Cas4s or Cas4. Now look at this. So this is one of the examples where 3 different Cas4 proteins are found in the same Archeal organism. So this is Pyrococcus yainosi, and we have Cas4A, Cas4B, and Cas4C. So if you look at first 2 Cas4s, they are all, so the blue, it is shown in blue, PyCas4A and PyCas4B. Both of these are surrounded by genes that are all associated with functions within Cas. So they all have Cas1, Cas2 similarity to proteins that have been involved in CRISPR function. But if you look at the third one, this is surrounded by proteins which have nothing to do with the Cas function. They are all kinds of enzymes. And so this is one way of looking at it. So if you look at the gene neighborhood of that gene, we were able to classify this as a solo Cas4. So this is what we mean by solo Cas4 and Cas associated Cas4. So anything that is occurring within the cassette near to other Cas genes is what is called as Cas associated Cas4s, and the others we are calling them as solo Cas4s. So this is just to give you a clarification. Now, if you look at it, whatever proteins we were able to whatever proteins we were able to gather from the bioinformatics thing, if you make a phylogenetic tree and you color them by their property of gene neighborhood and say whether they are solo or whether they are Cas4 associated, you see that they do divide into 2 different types. So if you look at this red and blue, they are more or less separated. They're not totally separated. So, but they are huge chunks, or they are huge clades that are pure blue or pure red. So if you look at the top, there is a pure red clade, and then it is followed by a pure blue clade, and then so basically that is just showing us that there are genes that are diverging from same ancestor, and that they are going in that direction. And so a lot of these organisms have the same gene, a copy of, or the homolog of the same gene, possibly from the same ancestor. And now, so basically, and then when we did this, we realized that whatever papers had been submitted so far, so there were 2 papers that had been submitted earlier, where they had shown that the possible function of Cas4 protein. So like I said, there were two structures, 4IC1 and 4R5Q, which were present in the PDB database. And so those, and they had done an elaborate study about telling us how they would be involved in CRISPR function. But ironically, when we do this analysis, it tells us that they are not actually Cas-associated Cas4s. And so they are solo Cas4. So we do, and they were showing so many differences among themselves that it was becoming difficult to comprehend how they would do a similar function. So therefore, this problem got solved a little bit. So we realized that whatever, so they are Cas4-like proteins, but they are not actually associated with CRISPR probably. And so there is no structure of Archeal Cas4 structure is known so far. And so therefore, we still need to characterize those. So if you look at it, we were able to see at least 4 of these here. So then these two, the ones 4R5 are both belonging to red. So this is solo. We started work on some of the blue sequences to figure out what we would do, what we could figure out from them in a later part. But otherwise, we continued looking at bioinformatics to figure out what if we could find some differences between them. So now, so for finding the differences between them, one of the techniques that we use is motif prediction, or looking for conserved motifs, which means that we are looking for amino acids that are highly conserved in the sequence of that protein. To see if it is highly conserved, then we realize that means that it would have an essential function inside the gene and inside that protein. And so we were looking at figuring out what are the regions that are similar between these two solo and Cas4 things, and what are the regions that are different. So we did this solo motif prediction from Mean software. And so if you look at it, this is what we get. So we did a motif prediction of this one. And then these are the regions that we found were common, which is expected because we would be able to, they're both Cas4, so they're actually doing some, they're orthologs of each other, they're homologs, they're distant homologs, and they have probably adapted to do different functions or become specialized for different function inside different organisms, but their origin remains the same, and so there is some activity that we expect to be common. For example, this, if you look at this CCC, this triple C remains common, this first C remains common between these two solo and Cas4 types. So there is some part of the protein that remains common, but there is some part that we call as differential motifs. So we take, so basically you can do it two ways. You can actually take one set as the background set, as a standard learning set, and the other set saying what are the motifs that are present in one but not present in the other one. So it is making a background and a foreground set, or say a standard noise and signal ratio. So therefore we try and figure out what are the amino acids that are present in one, but that are variable in one but are conserved in the other one. So is there a new function that a protein is taking? So all of those kinds we try doing differential motifs. So this is what again represented on the whole sequence of that protein, what we were looking at differential and common motifs, and I'll show you what this looks like on the structure. So this is what if you look at the structure of the protein, we have colored them according to whatever motifs we found, and then we were trying. So these are the regions that seem to be important for function, and these are the regions that seem to be having common motifs and differential motifs. So within those regions, these are the regions that we would want to mutate someday and kind of figure out what if the protein loses its function, if the protein becomes like, do they start showing similar activities? Do they start showing different activities when we mutate these ones? Now, if you look at it, this was a little confusing in the sense that we got with the signals that we were getting were not very clear. So they were mixed signals. The e-values were not very high. So then we decided that instead of looking at the whole tree and looking at all the Cas4s together, all the solo and all the Cas4 associated this together, let's look at a region that is neat. So in this case, the noise and signal ratio is very high in the sense that this clade has a clear classification. If you look at this has a clear classification of blue versus red. So we took only this part of the sequences and repeated the study, and which gave us better signals this time. So if you look at these will give you certain regions. So let's look at this one. So there's M3 domain here where it was showing. So there is something here, there is something here. These regions of the protein seem to be making a difference to the differential activity between the between the Cas-associated Cas4s and the solo Cas4s. So this was that. Then we also did, so because we are a wet lab, we are a structural biologist by training, so we went into figuring out what would be the activity of these things. And so basically for starting with that, or for starting for crystallography, we have to do cloning and expression of these genes. And so, which is, I think, pretty easy. And I think heterologous expression, RDNA technology is taught everywhere in class 12th and graduation. So I'm not going to elaborate on that. But if you have any questions, we could discuss those. But this is just showing, this picture is just showing you a strategy of how we selected the genes that we were interested in. So like I showed you a picture where we were seeing ABC, 3 genes from the same organism. So we looked at all of these. So we decided that we were going to clone all 3 of them and try to crystallize and characterize all 3 of them together to figure out what was the difference between, why does a single archaeal organism need 3 of these Cas4 genes and all of them, and what is the difference between all of them. And so if we study the difference between all 3 of them, maybe we will be able to figure out what their specific function is. So this was this. And then this is just showing you how these genes were cloned from amplified from their genomic DNA. So this is just a DNA gel, and you could see that these are labeled on the top. So these were the genes that were fluorescent. These can be seen here, different base pair sizes, and they were purified through gel purification. And then we are in the process of actually cloning them into DH5 alpha cells and PGMT vectors and other vectors. And so this is ongoing work, but we have so one of the genes we were able to work a little longer was coming from Pyrococcus yaenosi. So this is one of those genes. This is what the DNA looks like. This is what the sequence looks like. So this is about protein that is about 171 amino acids. And so we cloned it in PET28A. It gave good expression. So this was the expression in soup. And this was the expression, and then we were able to purify it. So you do a nickel purification column because it was histidine tagged, and then we tried to look at the elution, so we get purified protein here. And then so basically you can also do gel purification as a next step, or ion exchange chromatography as a next step. And so we were actually trying to do to see if the protein is actually showing activity in our hands. So one of the most important things is when you do a heterologous expression is whether your protein is folded correctly. And so the test of the basic test of that is whether you can actually see the activity. So if you see the activity, that more or less confirms that your protein is in viable form. And so this was the activity that has been reported earlier. So it was 5' 3' exonuclease and exonuclease activity, and we had already been characterized in other Cas4 proteins. So we know that this is what we should look for. But other than that, you can actually look for CD and fluorescence spectra of these native proteins to see whether they're folded correctly. So if you get a good peak at 280 and this, and you know that you see alpha helical content, so then your protein is folded correctly. And then we also see if it actually shows any changes over a wide range of temperatures. So we did both CD and fluorescence spectrophotometry to see what happens to the protein when we. So this, for the first 2 figures, basically tell you what happened. What does the graph of this look like when we do CD, circular characterization studies, when we heat the protein to different temperatures. So if you look at it, we starting from 25, incubating for 5 minutes at different temperatures, and taking the reading again 25, 30, 35 up to 90. And so so if you look at it, we see gradual changes. There is no abruptive changes here, and so therefore so basically it maintains its structure up to 75. And after 80, if you look at the first figure, after 80 it starts moving in the other direction, and so it basically starts showing differences. And so it is stable at very high temperatures, and so which basically says that the protein that we have in our hands is stable, and we could do assays at different temperatures and different conditions. And so it must be folded correctly. So if it is stable over a long range of temperatures, it could be an intrinsic property of the protein, but it also shows that that protein is folded correctly. Any protein that is not folded correctly or is will actually precipitate much faster if it is not, and the solubility will be very less. So this is what we did. And so we also tried again to denature it by using some kind of denaturing agents and guanidinium hydrochloride, which basically again says that it was stable to 2 molar of guanidinium hydrochloride, which kind of tells us that our protein is basically folded correctly and it should be in the functional form. So, so basically, and then we come back to our assays. So assays basically here are just exonuclease acids, right? So we are actually trying to cut off to see whether this will cut pieces of DNA. Now, we don't know what kind of DNA is it supposed to cut. So we know that other proteins have been shown to have exonuclease activity, but exonuclease could be, there are many different kinds of exonucleases, some that require a 5' overhang, some that require blunt ends, some that cut at the 3' end, some required to be annealed, some need splayed arms to be specific. So for that, you need to design different kinds of substrates. And so, so in this case, we actually take labeled oligonucleotides. So you are actually taking, these are oligonucleotides, 5' to 3', single strands, and you put a fluorescence label. So this is shown as Psi5 is shown as red star here, and this FAM is shown as 5 star. This green label is shown as green star here. So you make these combinations and then you combine them. So you anneal these sequences of oligonucleotides to form different kinds of, so you form different kinds of substrates. So now the different kinds of substrate that we have tried is obviously single stranded. So you want to see whether your exonuclease is working on a single stranded DNA or a double stranded DNA. Then if it is working on a double stranded DNA, is it working on a replication fork? So this is unlabeled replication fork. And then there is a splayed arm substrate, which is different from the replication fork. So here the arms, so this region is showing complementarity, it is sticking together, but these two regions are not. Then there could be a 5' flap region. So we expect it to cut here. So at this point, at the 5' exposed end, then it could also be a 3' exposed flap. So it could be cutting here, and then you could, so we have labeled and unlabeled versions of both. So this is just, the second row just shows you the labeled versions. So labeled versions, basically what you do is, if you incubate these kind of DNA with the protein, if they are chopped off, so you know that you started with 40 base pair substrate, but if you see them, if you see the fluorescence label if it is cut off here, so suppose in this substrate it is cut off here, your substrate will be, your end product will become 10 nucleotides, and so you will see them on the gel. So fluorescence label will exist at a gel at a 10 nucleotide base pair position. So therefore you will be able to figure out that, okay, there is a chopping place where at base pair 20. So this is, and again, this is, I guess you can recognize this is a Holder-Rigid junction, which is both at the kinds of things that we get inside the cells. So all of these kind of substrates were designed to see if which kind of substrate our protein of interest is recognizing. So whether it is working only on CRISPR type of things, single-stranded and then 5', 3', or it is working inside the cells or helping or has some innate housekeeping functions where when the cell is doing its own replication or has splayed arm or it has solid junctions and replication forks. Okay, so that is why we designed all of these activities, and then we did. So then you have to do an activity as say when you put the DNA substrate, you put your protein, you put metal ions and you do a reaction buffer, and then you incubate it for some time and you wait for the substrate to be cut to pieces, and then you run a native page for you run a native DNA gel also and figure out where it goes. So then so this reaction conditions were optimized. You need to optimize the temperature of the temperature and pH of this reaction because each protein works differently. So now if you look at it, we were able to see activity. If you see the red boxes at the bottom, so this is where we started. The black lines on the top is your substrate that is starting, and this is what is the cut substrate. So if you look at the red boxes, the amount that is accumulated there is darker and the top is lighter. So which basically says that you started with this substrate, but at the end of it, you get some cut product. And so the first lane is always the negative control, and the third lane is basically a protein that we tried with another tag. So instead of histidine tag, we were trying to use a SUMO tag because we were trying to increase the solubility of the protein, and we'll come back to that later, but that protein didn't show activity. So here the histidine tag is the center lane, the second lane is always showing you activity, and we tried it with different pH, different temperatures and different this. So some of them show. So the final thing that shows us activity is 5' exonuclease activity and 3' exonuclease activity at pH 8 and 55 degrees centigrade. So it doesn't show activity at higher temperatures, it doesn't show activity at lower pH. And so we can see activity only with the magnesium chloride as yet, and no activity seen in the presence of calcium chloride. So this is what we know so far. The other thing that we had wanted to do was to do crystallization. One of the drawbacks or the bottlenecks that we faced in this project was that although we had protein that was soluble and it was, but whenever we tried to crystallize it, so in the sense when you try to crystallize proteins, you need higher concentrations of that. So you have to go to about usually about 10 milligrams per ml, but the protein would start precipitating. So basically it became a bottleneck for us to be able to do crystallization trials. And so we tried using different tags. That is what people do when they're trying to increase the solubility. You try different buffers to make it stable. So we have these. This is the whole list of things that we have tried and not succeeded in still improving the stability or solubility of the protein. So we have tried different pH, adding different additives, lower induction temperatures, and adding different tags. So none of them have worked so far. So this is just showing you that we tried PET sumo vector. It is a solubility vector. It works very well for some of the other proteins in our lab. It has actually made a lot of difference to their solubilities. But in this case, we got the positive clones, we got everything done, and we tried purification of this one, and this sumo tag is so big it needs to be removed. So it was a long process, but then not every long thing actually gives you good results always. And so in this case, we didn't get any activity. So basically, if we remove the tag or without removing the tag, we did not get any activity on this one. So we had to. So once you get no activity on the protein, there doesn't seem to be a point in trying to crystallize that protein. So that is where we are at present. We are actually we are trying to optimize the solubility of this protein for crystallization. Although we can continue with our substrate assays to figure out what the activity of this protein is, and then we are trying to compare it to two other homologs that we will be doing soon in the expression in this one, and hopefully that put all together will give us some view of what this would look like. So basically, this is what our future holds. So we are actually cloning some few other genes. We will do the activity assays for the genes that we have designed substrates for. And crystallization trials, obviously, we are not giving up. And then some biophysical analysis will be done to figure out this, to see what, and some mutation studies will be required to see what, once we do, figure out what activities they are. So then we make some mutation studies and figure out how do we lose, which residues are responsible for 5' activity and which residues are responsible for 3' activity, and so on. So those are basically things that we will do further. This is some of the reading material that we have used earlier and what other people had done so far. And this is basically just my acknowledgement. So there were 3 people, PhD and MPhil students who have worked on this one. And there is one research associate, Dr. Muskan Jain, who's currently working on this. And we have a collaborator in NIPGR Delhi, Dr. Vineet Kaur, who's helped us with the exonuclease assays. And then, obviously, thanks to the funding agencies who have given us grants to do this, and my own department for this one. And thank you all for listening. I am ready to take questions. I hope I gave you something new to listen to. I hope this was something different than what you've learned before, and it added some value to the Science Day celebrations today. Yeah, sure. So Monica's raised the hand. Can I go ahead and answer questions from Monica?
Yeah, hello, Dr. Manisha. I hope
Yeah.
you have recognized me. I once came to you. I'm one of the faculty in Miranda
Hi,
House.
Monica. Oh, right. Now I do. Sorry, sorry. I'm sorry. I couldn't recognize you by the name, but by face I do. Yeah.
Yeah. So, Dr. Manisha, very interesting work. And
Thank you so much.
I just want to know, because we are working on MTB, have
Mhm.
you found any Cas or Cas associated genes and their activities in MTB? Because I'm really unaware of it.
Actually, it has been found in at least 45% of different bacteria. I'm sorry, I can't answer that question directly because our lab has always been working on Archeal systems. We do that for a variety of reasons because we actually started as an Archeal lab. And so I am not sure that I haven't looked at MTB at all. And so I have no idea whether these are present in MTB, but they could very well be. but they have been seen in 45% of all bacteria. So, but I could look at it and come back to you. I mean,
Sure,
unfortunately, I don't
I
have an
will
answer
get
just now.
in touch with you. And one more question, Dr. Manisha.
Yeah.
Like this CD analysis which you have done,
Yes.
cellular
Yes.
dichroism.
Mm-hmm.
So in those in those essays, what
Mm-hmm.
buffers and all you work with, because we are also working on it and we are not getting any success because probably the buffer is the problem or
So
protein.
in CDs, in secular dichroism studies, you have a CD instrument that
No,
you're using.
we come to your facility only.
Okay, so basically, if you come to our facility, then our facility is working fine. So I can assume there is some problem with. So sometimes there is a problem with the protein. I don't know whether you've tried multiple proteins or multiple homologs, but usually you could look at our papers that we have published in the lab. If you come, I could put you in touch with one of the students who does CD.
Yeah,
So
sure. That is what I want
yeah,
actually.
yeah, perfectly fine. That is no big deal at all. So is it so? So you can actually come and. You can come and look at that here, but then we do CD very often, and so if you look at our papers or a PhD thesis, I think the buffers will be very detailed there. Each protein requires a slightly different buffer in which they are stable, but usually it is low molecular, low p, low ionic strength buffers
So
with low
that
salt.
means sodium chloride, because my major worry is because we
Hmm.
generally use normal phosphate buffer saline. So
Yes.
is it important that we reduce the sodium chloride
Yes, I think
concentration?
so. Yes, I think so. So that may make the difference this one, but your protein has to be the fight is between trying to stabilize the protein and between trying to, so that is what happens with our protein also, that we get but CD requires very low concentrations.
Yeah.
So that's why it is still able, so we are still able to do the CD assays, but our protein is not stable if we remove NaCl. And then that is why for higher concentrations, even higher concentrations of NaCl are not helping it go beyond 2 milligrams per ml. But CD is still doable. So each protein will behave differently, but we can all be, I think, we can always look at your buffers and see
So,
what you have tried already. And if you can suggest something else, you are more than welcome to
sure,
anytime to
I'll
please
be in
stop
touch
by.
with you.
Yeah,
So
stop by. We
thank
are
you,
just
Dr.
next door to
Manisha.
CD, so we are just opposite.
Yeah.
the
Thank you,
CD.
Dr. Manisha. So I would not be in between you and students. So
Sure.
thank you so much.
So there were some other students.
Yeah.
Yes, there was somebody who was asking whether lyophilization can be done. So basically, lyophilization is a technique where you're drying the protein, and lyophilization basically turns it into a powder. So lyophilization is necessary or useful when you're trying to store the protein for a long time. But lyophilization actually, because sometimes most proteins. For most proteins, lyophilization is not a good technique to even store them, because it actually dehydrates them. And once you add water, not all water will find the correct place, and it may actually distort the structure of the protein, which may lead to loss of activity. And what we are trying to do the crystallization for is to determine the structure of the protein. The crystallization is totally different technique from lyophilization. Crystallization basically involves arrangement of protein molecules in a long array, so that you can actually throw X-rays on them, so that they diffract the X-rays, and then they diffract the X-rays, and we are able to use that diffraction pattern to determine the structure of the protein. But that cannot be done in powder, especially because in powder all the molecules are oriented in a different orientation. So powder is basically a heterogeneous mixture, and what we need for X-ray crystallography is a homogeneous arrangement of protein atoms. So every first protein has to have a second protein arranged in the same conformation behind the other, so that it makes an array. It makes a whole array, and then only you can see this one. I hope I have answered your question. The person who had asked about lyophilization. Does that make sense to you now? Lyophilization versus crystallization. If you have anything more, and if it doesn't help, then okay. And then I think there was another question which I saw, which popped up, which said, can we do AFM? Of course, we can do a variety of things with this. We can even do NMR other than crystallography, but then There is practical limitations, like I have to find a collaborator who's ready to do this. And so we are basically trained as crystallographers, so but these are techniques that take a lifetime to kind of master. So basically you have to have a PhD at least or you have had to work on this for about 5 years or something to be able to do something independently. So it is not easy to kind of just jump from one technique to another. So there are options, yes, but whether they're available for me is the limiting factor. We can always have a collaborator. So basically, it means that the same person it's very hard for same person to have so much expertise that in different technology, that you do crystallography also, otherwise you do AFM or electron force microscopy also, and you can do NMR spectroscopy also. So basically, these are techniques that require years of learning. So you become an expert only about after spending 5 years in each technique. So then we don't have that kind of lifetimes. And so basically that means that I cannot do this on my own, but we can always find a collaborator. Yes, we can always find a collaborator, and this is doable, but we have not been able to do it so far. And sometime, and for crystallography, it is kind of given that it will take time to crystallize, and there are other techniques. So we haven't given up on crystallography yet, and so we haven't really started looking at collaborators yet. Anything else? So there was somebody else who was asking another question. I hope I've answered the question. If you have a continued doubt, or if it doesn't answer your question, please let me know. Should I look at the chat box to figure out? Okay, there were only two questions. So Nikhil's question is more or less answered. Okay, so they're both the questions from Nikhil, so we are good. Anybody else? We finished quite early, I guess, right? No more questions? I hope I made sense to somebody. Okay, so is there anything else you would like to ask?
Ma'am, I don't see any more questions in the chat box.
Okay, so I hope I hope I gave you something to think about and something new to learn. And I'd like to thank you all for this great invitation. It was good. It was a great initiative from your part, and thank you for the invitation. So
Thank you so much, ma'am.
Really
I would
thank you.
request
great
Chaitanya,
comprehensive
the
lecture.
General Secretary of SYNAPSE, to deliver the vote of thanks for ma'am.
Oh, okay. Thank you.
It's my privilege to propose a vote of thanks on this occasion. I, Chaitanya Sunil Bharadwaj, General Secretary of SYNAPSE, on behalf of the entire union, would like to thank our speaker, Dr. Manisha Goel, ma'am, for gracing this event and sharing with us your valued study on the CRISPR systems. Thank you, ma'am, for giving us your precious time and for your cooperation. We are all inspired by your words, and certainly we have got new subjects and topics to study on in the future as well. You helped us learn a lot about this technology in detail. I would like to take this opportunity to thank our principal, Dr. Vijayalakshmi Nanda ma'am, for providing us the opportunity to organize this webinar, and Dr. Nisha Vishist and Dr. Yasha Yadav ma'am for their support and guidance. I would also extend my thanks to the president, vice president, event managers, and all the other union members of SYNAPSE for successfully organizing the event. And last but not the least, I would extend my gratitude to the audience for their active participation.
Thank you, everyone. It was great interacting with you, and thank you so much for the invitation. Thank you, ma'am, principal ma'am, and other organizers, and thank you, students. I hope you enjoyed the talk, and take care. Wish you all the best, everybody.
I would request the participants to stay on the meet and fill the feedback form for the certificates.